Okay, just wanted to provide a quick video that can help you to approach reading this paper. I'm just afraid if I don't give you something, you're going to start reading this paper and just give up. Right? You're not going to know which parts to focus in on and really get any of the reward from an understanding of the paper. Some parts of the paper, honestly, I don't really understand well, even though I'm a first author on it. That's why we collaborate with people. My colleagues, Jessica Wooten, who's at Piedmont, and Todd Pearson, who's now down at Kennesaw, did a lot of the lab type of work here. Carlos is kind of old school biologist. He understands the lab stuff, but he's really been involved with a lot of these species descriptions. So just want to kind of be sure to read through the abstract. That gives you a really good broad overview of the paper and the study, and should help you to kind of orient yourself as you read through the paper. And again, the introduction, right, beginning here gives us an impetus for why we did the study, about describing species, gives you a little more information about specific description of species in amphibians, getting even more specific to this genus Plethodon. These are lungless salamanders, really reach their highest diversity here in the eastern U.S. And some of the challenges of the taxonomy and phylogenetics of the group. And then we're really getting more specific and looking at this specific group, saying specific a lot, this group, the Whirly salamander group. I would spend some pretty good time on these two paragraphs. And what I would do is write down the name of these groups, right? I'm saying groups being Plethodon whirlii, Plethodon jacksoni, the cow-knob salamander P. punctatus, P. dixi, and then the yellow-spotted Whirly salamander. Because these are the members of this species group, and we're gonna keep revisiting those groups as we go through. So now we're just introducing them. These are going to be the main players here. The next couple paragraphs describe the status of the state of knowledge when we started our research. It talks about some of the previous research and fairly recent work on the group, trying to make sense. Really, and the reason that we're talking about this paper is because I find it's a good example. One that obviously I can communicate well because I've lived through it, but is, again, here's a bunch of vertebrate animals. There's morphological diversity. We have geographic diversity. How do we make sense of them, that group, and how do we categorize and put them into species groups? And this is an example of sort of at the beginning of this. It's kind of a mess. Frankly, there's a lot of issues coming out of Kuchta et al.'s research, and that's where we kind of stepped in and made some order out of the chaos. So spend some time with the intro, getting acquainted with the work, materials and methods. Here, look for a couple things. One is, what are the kind of details that are included in order to make this a repeatable type of study. That's a main premise of science. Is it needs to be repeatable. It needs to be we need to be able to validate somebody else's research by doing the same work and same type of experiments and coming up with the same conclusions. Okay, so the first part of the methods describes how and where we collected animal tissue from and how that fits in with those 5 groups. You can read through that. This table these are the actual each of these specimen IDs. This was an animal collected from Letcher County, Kentucky. This is exactly where. and then this tells us where you can find that actual genetic sequence on GenBank. Okay, so there's all the animals, 65 of them. This tells what we did with those tissues, how we extracted DNA. We did 2 different types of genetic analysis. One looking at a specific gene called Site B, which is a mitochondrial gene, and we're looking at 650 A's, T's, C's, and G's within this one gene. Okay, so that gene codes for a specific protein used in respiration, I believe, and we sequenced the information contained in part of that gene. And then we also did a kind of a new approach to making to phylogenetics, which is using whole genomic data. And this is was Todd Pearson's real contribution, making these what are called 3-rad libraries. Read over that. I guess for me, the big thing I want you to understand there is the difference between using a single gene and using a genomic approach, the strengths and weaknesses of the two, and more so than the specific details of using pip and preps and preparing libraries. Honestly, those are the things that I am pretty vague on, and that's again why I work with experts on that. This shows spend a little time with this figure, match it up with the collections. This shows where tissues were collected from and how that fits into both the Site B single gene approach and the genomic analysis, because the genomic analysis were done on 20 individuals, which were largely a subset of the 65 individuals. So we collected 65. We used the tissue from them to make a tree, okay? And then based on that tree, we chose a subset of 20 individuals to do the genomic analysis on, okay? And this will help you to piece apart how we chose the subset and where those subset came from, okay? More just, again, details on making the cladogram phylogenetic trees from the genetic information that we got from the tissue. Another thing we did was look at the shape or the morphology of these groups. Those, again, going back to those same five groups. This tells how we measured, what we measured. This is real similar to the ANOLA lab that many of you guys did in General Zoe. These are just straight line distances, width of the body, the width of one individual eyeball or orbit, distance between orbits, distance between nostrils, all this suite of variables that we measured. This tells you guys exactly which specimen. So you could go to these museums, request, go to the Smithsonian in Washington and request specimen 143190, get that animal, do a measurement of all of these variables, and you should get the same answer that we did. Again, repeatability. Okay, so this is the morphometrics, and then that's really what we did. We collected animals, we extracted DNA, sequenced one gene, we built libraries of genomic data, we built trees, we measured the shape of the animals, right? And then that brings us to the results. Okay, so here, sort of the approach that I'd like for you to do. This first part, the genetic and genomic data. Okay, spend some time and again match up those groups from the intro. When I'm referring to groups, in this part of the results here, should be the story continued about those 5 groups, and then take the description here, really what this is describing phylogenies or trees. Okay, look at the verbal description of the tree and then match that up with what you see in the actual tree. Here's figure 2. So that first paragraph is largely a verbal description of what is going on in this tree. Remember those the big table that I showed you, and it had the different specimens. These are the same specimens, right? So this is one individual, an animal collected Tennessee 4. We can zoom back up to the table. That individual was collected in Campbell County, Kentucky, right? And this is where you find that sequence. Okay, this is where it falls out in the Site V phylogeny. Okay, so those are all individual whirly salamanders. This is the description of that tree, right? And then you're going to see another description of figure 3 and 4, which shows the same type of tree but generated from the genomic data. And we see again here those same 5 groups: yellow spotted whirly-eye, northern whirly-eye punctatus, jacksoni, dixi, and then southern whirly-eye. So read through that, match up with what you're seeing here with what you're seeing in those figures. Compare the two types of trees and see how similar and dissimilar they are. The morphometric data, basically the story here, is simply says that the groups are different shaped, and that if I tell this computer all of the measurements of those measurements from that table, so if I feed it into a computer and for a given individual salamander, and I tell it its tail width and its length of the hind limb and distance from the snout to the cloaca, if I tell the computer all of those measurements, it will be able to predict which one of those 5 groups that individual belongs to. Okay, so what that tells us is, of course, that these groups have different shapes. Okay, so those are the main results: the phylogenetic information and the morphometric information. And when you read through those, right, that leads us into the taxonomy part of the paper. Okay, this is still part of the results. And so that is, again, the main point of this paper, why I want you guys to read it, is, right, we have a group, collect the data, we get the results, and then we use those results to inform how we group and name members of this species group. Okay, so considering that the groups are distinguishable based on their genetic and genomic data, and considering that they are different shaped, how do we group and name these salamanders? Okay, and so you can read through this taxonomy. This just starts with the introduction to that very question I just described to you. And then it goes through and it says, okay, considering all the data, right, considering that if we look at the genomic data, that all the yellow spotted whirly-eye are on one long branch that is very distinctive from this branch that contains northern whirly-eye and punctatus, and then we see this other branch that contains southern whirly-eye and jacksoni, and then this other branch that contains dixi. We recommended the recognition of these species: P. punctatus, P. dixi, P. jacksoni, P. whirly-eye, and then this is a new species, right? That before this paper didn't exist, and we are proposing this as a new species, species new to science. Okay, and so I segued to this reading from the last reading. We talked about species and how to define them, and then really this paper is an example of how one of a new species is named and becomes known to science, becomes part of the catalog of life. And so this portion of the paper, up to this point, is providing evidence for the idea that those yellow-spotted whirly-eye group is a new and distinctive species. And this part of the paper basically is doing the job of describing that new species. And so you're going to see these are standard. These are the standard parts of a species description. We have to define one individual organism that exists in a museum. In this case, the museum is in Berkeley, California, the Museum of Vertebrate Zoology. And if you went there and asked for 291261, you will find a female individual that I collected in 2011, and that serves as the archetypal Plethodon pollyi. That individual animal, in fact, you will see that that's her right here. This is Plethodon pollyi. This is the type series, and if you want to know what one looks like, this is the comparison. Right? So these are the paratypes that were collected that same day. Diagnosis tells us how to tell Plethodon pollyi from every other species of salamander known on Earth. This tells us how it is unique and how to be able to diagnose it as being Plethodon pollyi. Okay, this is a description of that individual animal. Here she is in life. Again, this is her. See the same spots. Okay, this is part of the. This is another Plethodon pollyi to show variation in the group. We can still see some of the spots on the back. In fact, I believe that is this individual here, and then to show part of the diagnosis is the difference in their ventral coloration, and so I've showed that in this picture. Talks about variation. So, well, although there is one archetypal Plethodon pollyi, we did make sure to describe so that there is some variation, because we know that within a given species, right, there is variation. But that variation, so individuals of Plethodon pollyi should all be more similar to each other than they are to members of, say, Plethodon jacksoni. Okay. Description of type of habitats they use, where they're found. Etymology is description of basically the etymology is a study of words, and so this describes where the name comes from. So this pollyi is named in honor of my mentor from my time spent at Marshall University, Dr. Tom Polley, and this just lets people know where the name came from and honors him. Some more results that are kind of peppered throughout here, and then that leads us into the discussion, and spend some time on the discussion. It kind of wraps up. Again, we're starting with disorder. We have these groups that are seem to be different, but there are indications they're different, but why they were not raised to species level. That leads into our study and how we aim to fill in those gaps. Then the methods at how we did that. Results indicate, what we found in our genetic analysis and our morphometric analysis. Then we talked about how that can be used to revise the taxonomy of the group. We go through that actual specific taxonomy. We propose a new species, and then the discussion is just kind of the wrap up. So make sure to read through that. And this is an opportunity for us to, talk about where we see the science going from here. The science in terms of this group. There are still unresolved issues in the Plethodon-Whirlii group. We feel like we left it better than we found it, but there's still some unresolved issues. Those are described here in the discussion. And that's how science works, right? What's known, contribute your knowledge, and then at the end say, okay, all that considered, here is still room for growth and pushing the envelope further. And really, so when you want to find something to research, a lot of times I'm going to go to the discussion section of the paper and say, okay, here are things that have been suggested as fruitful areas for research. So I'm hopeful, again, this kind of give you some a foothold into understanding this paper. We'll discuss it next week. I'm going to put up an assignment here on Canvas that will ask you some questions about the reading and get you to delve into it a little bit more deeply.